Now, I mentioned the word innovate, and speaking of innovation, I have to say it is my pleasure to introduce our keynote speaker for the morning. I would like to introduce Dr. Sue Behrens. I've known Sue since the mid-1990s during her time at Merck. And this morning we have limited time, so I won't be able to go into any detail into a host of great stories I could tell you about Sue. So we'll have to do that some other time, maybe telling about maybe when Sue and I were recruiting at the University of Michigan together, or some of our experiences at MMD leadership conferences. But what I will say is that Sue is a wonderful person and a great engineer. But since you haven't known her since the mid-1990s, let me tell you a little bit about her. Sue earned her bachelor's degree in chemical engineering at the University of Michigan and her PhD at UC Berkeley. And at Merck, Sue held a variety of different progressive positions in MMD. She started in technical operations, supporting sterile API and fermentation processes. She was in MRL and GES doing process design. She oversaw sterile process technology and engineering functions at West Point, and was the senior director of biologics manufacturing science and commercialization when she left Merck to join Integrated Product Solutions in Blue Bell, Pennsylvania. Since 2019, Dr. Behrens has been the George B. and Joy Rathmann Professor in Bioprocessing and Director of the Amgen Bioprocessing Center at the Keck Graduate Institute. She's done that while having her own consulting business in biologics. As I said, besides being an expert in biotechnology, Sue is a wonderful person and a good friend. And I am thrilled to hear her talk, a talk entitled The Cycle of Innovation, Advanced Biomanufacturing and Single-Use Technology. Colleagues, please welcome Dr. Susan Behrens. Sue?
Hi, everybody. So good morning. I will get started here. And I thank you, Mike, for that wonderful introduction, as well as thank you for inviting me and allowing me to come back to Merck. I was hoping we would all be together in Pennsylvania, but here we are today. This is how we're teaching. So this is something that I am used to, and we will get started. We should be sharing. And I will go to full screen. So I hope that you all have been able to get used to this as well, this new technology that we work in. And I wanted to take a minute here and—
Sue, sorry,
Yes.
John, 1 second. We're seeing your presenter mode. If you can go to display settings in front of you in your presenter mode and just swap
Okay,
displays.
just a second. Actually, let me just switch screens that we're sharing then. Is that, so now we should be in the right, is that better?
Yep.
All right, sorry about that. Technology. So one of the things we do when we are meeting with our students is we try to take advantage of their input as well. And I'm not going to do a lot of that today, but I just wanted to give you guys one chance to see one of the tools we use called Pool Everywhere. Works great, integrates directly into PowerPoint, so it makes it easy for me. I told my kids a long time ago that although I'm a process engineer, what I really am is a PowerPoint engineer. So I would go ahead and sign in at any time. It'll stay active the whole time I'm talking. So if you go to polev.com/Sue Behrens 254, you can see people starting to drop their little spots. So we can come back to this as we go. And you can see, as expected, a lot of you are there in the East Coast. That's me, the lonely little dot out on the West Coast. And I think we have a few people who might be joining us. There's somebody over there in the Netherlands. So welcome. So I'll come back and we'll look at where we collected our spots when I come back. So what I wanted to talk about today was what I think of as a cycle of innovation. So for me, thinking about advanced biomanufacturing as I've moved into academia, I've moved away from the day-to-day of getting products made, or when I was at IPS, getting facilities designed, and thinking about where are we going? Where is this industry going, and how are we making advances? And when we talk about advanced biomanufacturing, and I'll talk about a description of what I think that is in a minute, I really think it drives, it's the mother of invention, drives us to single-use systems. And then those single-use systems also enable us to do advanced biomanufacturing, which wouldn't be happening at all. So I think we're in this cycle, and I really want to, kind of tie back to what Mike said, that innovation is the key to moving us forward. And even though we might not know directly where we're going, if we keep pushing forward, we keep driving advances, and we allow things that we didn't even envision 30 years ago. I think Mike and I can both tell you when we started in this business that we had no idea where we would be today. And I look forward to where we will be in 10 or 20, 30 years, providing medicines and medical treatments to people that we couldn't have imagined 50 years ago. So I'm going to just talk real briefly about what is KGI and the Amgen Bioprocessing Center, talk a little bit, about, like I said, about what I think of as advanced biomanufacturing, and then propose a framework for innovation that comes out of some work that was done around advanced biomanufacturing. And then I'll tie back together single-use and advanced biomanufacturing at the end, and then certainly ready for some questions from the audience. So, as Mike mentioned, I moved out here last summer to Keck Graduate Institution. We're located in Claremont, California, which is the easternmost city in LA County. So we are out here with fires and earthquakes, a new kind of challenge for me. But I came here because of the mission of this organization. So it was a big move and a crazy year to make a move across country. But what we do is so different and unique than anywhere else that I've seen. And I really was encouraged and excited to be here. So the whole mission of KGI, which is a relatively new college, founded in 1997 by the president of Harvey Mudd, because he saw a gap in taking discoveries from life science and being able to translate them into therapeutics that would provide benefit for society. So we offer a MS program or an MBS program, Master of Business in Science, which is like an MBA, but we focus on science and not on case studies in hotels or airlines or sneakers. And we offer PhD. We have a school of pharmacy that offers a PharmD, an occupational therapy, and a PA program. We're in progress of starting a school of medicine, which is targeted for 2024. And we just got some really exciting news this week where we have a major founding donation to help that get off the ground. But really, the thing about KGI that's different is the entrepreneurial culture and the industry collaboration, which made this a natural fit for me and made me excited to be able to tie our talent for the future generation to the industry of the future as well. So the Amgen Bioprocessing Center within KGI is my role, my responsibility, and we offer instruction across all of the different topics in biomanufacturing. So we're focused on bioprocessing. We were founded by a grant from Amgen in 2003. The founding director was Matt Krogan, who many of you probably know from his time either at MIT or at different consulting around the industry. I would say he's a giant in our industry. We also recently benefited from some part of Shamblu, who spent about 4 years here and helped get the Masters of Engineering program off the ground. And we also do a lot of research. So we all as faculty have research. I have my first PhD student, I'm proud to say, and we're working on a new project and have a connection to Merck through her committee. And we bring industry into everything we do. So we have an industry sponsors for small research projects. We have industry coming in to talk to us and provide support. And we're really in a manufacturing opportunities to build new partnerships. One of the things that we do is what's called the Team Masters Project, and we collaborate with industry to build a new projects and new talent at the same time. And they're really exciting projects. You can see here to the right, that's our lab at the top. We also have our annual conference and advisory board meeting. Many of you may recognize Vic Vinci there. He's the chair of the advisory board right now, and looking at a poster with some of our graduate students who've put together their research to share. So what is advanced biomanufacturing? So I want to start with talking about what's advanced medicine, because I think the two are interweaved. So there are three pillars of medicine that we're starting to talk about, and it really came through this publication by came out of UCLA, and thinking about our growth as a medical industry. How are we providing therapies to patients? And we started over here on the left in molecular therapies, and that's really, you know, APIs like Crixavan that we mentioned, thinking about how we grew into bigger and bigger molecules, but we were still at the molecular level, looking at proteins like monoclonal antibodies and others, enzyme replacements. And then we moved into thinking about gene therapies. And what if we figured out how to fix the genes in our systems that aren't working right, and started thinking about adding and changing the genomics. And then we moved into cellular therapies, where we're actually treating cells and getting cells to do what we want them to do, rather than not operating properly in our system. And you can see down here, vaccines is down here, kind of in between, because it's considered a cellular therapy in that you're trying to stimulate the immune cells in vivo to create a new response. And so, although I would say that this is not chronological, because as we know, vaccines are probably the first biomanufacturing product, we do recognize that they are a cellular therapy. And a lot of the technologies that we use for vaccines are being translated into these cell therapy products. So another way to think about advanced biomanufacturing, and that comes up if you start to look in the literature at all, is it's about new regenerative tissue. So here we go. So we went from talking about the molecules to the genes to cells. Now we're talking about tissues and organoids and the ability to make new systems that can treat the body's dysfunctions. And so you can see here a representation by a very esteemed group of authors that came together from all over the world to talk about what do we need to do to achieve those goals in therapy. And you can see here this idea of a tissue foundry. How do we build and get to a tissue that's going to work? And kind of coming off to the side are our traditional proteins and small molecules. And what I would tell you is that it's called advanced biomanufacturing, but everything that's listed here are actually products, organs on the chip, tissues, desired volume, and the manufacturing is in the arrows. So what we focus on and what we do is really represented in a lot of these conversations, very minimally with the arrows. And so I want to talk about that space, because I think that's what's important to us as we move forward as an industry. So so moving into talking about that manufacturing, we talk about protein manufacturing and how we got here with single use. Really was driven out of the protein space. And you can see this is from our friends at Millipore Sigma, great resources. And they're showing here a future that gets us past 2025, where we start with our standard MAB template in 2017. And that one actually starts with single use because it was 2017. Of course, this started in stainless steel in 1970 something. But we're now getting there, and in single use, we have a full train in single use. Now we're starting to look at what's the next step. in advancement. What are we going to do next? Well, we're going to look at process intensification. We want to be able to get more out of less, which is the definition of intensification. And we start that with cell line development and trying to get cell lines that make more out of each cell. And then we talk about how do we get that in our equipment? How do we get smaller equipment that makes more? And that drives us towards continuous processing. And you can see the vision that we go straight from a bioreactor into a vial in their 2025 vision. Gene therapies are another area where we're looking at advancing biomanufacturing. So the cells are moving, we're taking from patients and delivering those cells to the manufacturing facility where we modify them, expand them, and send them back to the patient. And then we also, as part of gene and cell therapy, we talk about this concept of allogeneic versus autologous. So where do those cells come from? If they come directly from the patient, we call this autologous. And we need to do some transformation of those cells and send them back to that patient. If it's allogeneic, we get a cell bank, which starts to sound a little bit more like what we do with our traditional biomanufacturing. And then we modify those cells and then we can send them to multiple patients. And there's a lot of advantages to allogeneic treatments in that we're taking cells from healthy individuals rather than sick individuals. And then we have a strong manufacturing opportunity for cell banks. Another thing to think about, like I said, is tissues. So how are we generating new tissues? And this is an example of modifying tissues and retransplanting to patients for mesenchymal cells to generate a new and safer response for cancer treatments or for regeneration of damaged cells, for example, in the heart after a heart attack. But I just want to point out that these cells steps look familiar to us. So before I leave this most advanced biomanufacturing, these cells really do look, this process looks like what we do. So we have the Petri dishes, maybe is a little bit small, but we go to a cell bank if it's allogeneic, we expand on a tea flask, and we move forward. And I will say that we talk about single-use manufacturing and advancing manufacturing, that upstream in the biological cell growth stage, we've certainly been using single-use systems for a long time for vaccines. Roller bottles are single-use systems. Nunc cell trays are single-use systems. So we've been advancing biomanufacturing and generating a deep sense of knowledge for a long time that we can apply in this space. So we think about advancing manufacturing, how do we think about all the steps we do? So in cell growth, we think about our traditional steps of seeding, but we need to get to large numbers of cells very fast when we think about cell therapies or tissue regeneration type therapies. We need obviously very high reproducibility from completely different starting materials when we talk about autologous cells. And that's a really tough challenge that we really need to take all our learnings together and figure out how to bring that forward. We also are still thinking about adherent versus suspension cells. So we can get to suspension by using microcarriers for some of these steps, and we're starting to really make a lot of progress in that space. The cells, though, are going to require adherent systems, and we find a lot more sensitivity to what that adherent system is. The cells, for example, when you're growing bone cells versus growing endothelial cells, they want to be on different surfaces. They want to have different levels of stiffness to grow on in order to get to each other. So we're learning and trying to figure out how to do that as we advance biomanufacturing towards these advanced medicines. Bioseparations, challenges there. We're starting to think about how to separate cells from each other. Not something we typically do in our current environment, but we're looking for which cell got the modification we wanted it to get and is ready to go to the patient. We're looking for how do we separate viruses from viruses. So we need viral clearance on our viral products. So we've got a lot more activity going on in trying to understand and adapt the bioseparations technologies that we have to these new products. And then finally, product delivery. Everything can't be in a vial or a syringe anymore. We really need to get into more advanced delivery technologies, including things like like transdermal patches. Delivery of organoids is obviously, it's an implant type system. So we need to be thinking about how do we advance manufacturing across all parts of our business. So another piece to think about in advancing is the steps in the overall production and system. So it's not just the technology platform, it's the production system. And if we don't adapt our entire production system as we move the technology platform forward, we won't get the value out of that technology platform. And so what you see here actually is a representation, a slide that was presented by Tony Marius-Luce, who's at AstraZeneca. And they're really progressing in their technology, moving forward in the Pharma 4.0 space, digitalization, driving digitalization everywhere, getting our information into one place and being able to eliminate a lot of these manual steps that cause error and really constrain us from moving forward into this vision we have of ongoing continuous product that meets the highest level of quality. So I'm going to switch gears now and talk about a framework for innovation. And really, I was inspired actually by the talk, the paper that I presented earlier for Tissue Foundries, to start thinking about how do we move innovation forward? And thinking about that, I've pulled out of there 3 main tenets that I think are really important. Learn from biology. This is actually the tenet that motto of Jay Bailey, who was Bailey and Alice, that all of us biochemical engineers started with his text. And we really need to go back to thinking about that. We've spent our time thinking about learning from biology, meaning let's figure out and map the genome and figure out which gene we can move, to we really need to understand cellular processes. We really need to dive into and expand our knowledge of how cells work and how we bring cells together to make the products that we need for advanced medicines. Rapid prototyping. We need to get from an idea to a demonstration quickly. We need to be spending our time demonstrating and implementing our technology rather than thinking about our technology. And then from an implementation point of view, we need to take that demonstrated science and quickly turn it into a production facility. So we need to go from a lab-based concept into a production facility. So I'm just going to talk about each one of these briefly, and then we can come back to the single use and how this connects. So in terms of learning from biology, a couple of things that I'd point out. Biology, by definition, is an integrated system. Our cells are systems based. There's a lot of monitoring and feedback that's built into our systems, and those naturally cause and allow for the highest quality operation on a regular basis. Our cells are efficient. We are continuous organisms. So I always tell my students that we are the first moving fed batch system, and we are really efficient, and the cells are operating in a continuous fashion, as well as being very productive. So if you think about intensification and what's happening inside a single cell, it's really remarkable. And the other thing that I think to learn from biology is about recycle. We do obviously generate waste when we do any kind of biology, but a large amount of a cellular material is recycled and used and pushed forward. And we really need to think about that in sustaining our environments. So for an example of that, I'm going to show you this picture. This is the Roche metabolic pathways diagram, and it is huge. So I just cut out a tiny little piece of what's happening in the cell. And you can see how complex this is. And they don't need a Pharma 4.0 to manage this, right? Our cells are doing this every day, all day while we sleep. And what we look at this, it's really a bunch of building blocks they put together. So it's built out of smaller blocks that allow us to succeed. So it's really thinking about those blocks that allows us to then say, okay, so if I control this block and then I control the feed to that block, then I can build larger and larger and larger inside this one unit cell. So thinking about design and prototyping, that building block concept comes into that. So if we learn from biology that we should do things in building blocks, it helps us to see how do we quickly move technology forward. So if we want to build a new single-use bioreactor in 1992, we would have wanted to think about the agitation. We would have wanted to think about the sparging. We would have wanted to think about the sensing and the addition of acid and base, and how did we bring sterile media in? But if we thought about that all as one unit, it would be overwhelming. So what we do is we chunk those off and think about them one at a time. And I think that that's a really important thing that moves innovation forward. We get stuck when we try to think about achieving it all at once. We need some sort of definition of a building block, and that's what I would consider standardization. And I know as an industry, our vendors are really helping us move that forward so that we can connect to each other. We want to think about what's high throughput design and prototyping. So we're getting there, and this is something that we can take advantage of other industries. How do we miniaturize things? The smaller we can make these building blocks that still reflect what we're doing at scale, the faster we can move through the design, build, test, learn cycle and make sure that what we're making is going to work as we scale it up. And then another thing to really think about is information exchange. It doesn't do any good to have an idea and only keep it in one space. So we need everybody talking to each other, discovery people who have the ideas, developers who know how to translate that idea into a full scale, end users and manufacturers who are going to actually implement that technology, the vendors who are going to build it. It has to be one community. And I have to say, that's one of the things that the single-use network has really always done, is drive that connection between all of these users. And then as an example of that, I'm going to talk about COVID masks. So what happened right away when we got into this space in the with COVID, and we all needed masks and no one had masks, where were we going to get this PPE? And over spring break, we shut down on the Friday before spring break. And over spring break, one of our professors, Anna Hickerson, who is the program director for the medical device engineering program here at KGI, went home, and this is her in the background there on her dining room table, took home a printer and a vacuum molding device, and came up with this prototype and started making masks based on a design. So she went straight from ideas to prototypes, didn't stop and think. So she's about, oh, I can't do this, it's going to take too long. She just started doing. And I think that's really important, is that we start thinking about. And she did this under that concept of building blocks. So she started with this design on the left. The bluish shade is kind of the support structure. You can see what's in the white is the actual filter. And you can see that as she molds the support structure in the vacuum molder there, and then adds the filter, she gets the finished product, and very rapidly is able to make many of these. And on the bottom is a similar thing, also masks from COVID. But you can see this came from University of Wisconsin, Milwaukee. And when they documented their process, they showed us, they looked at all these different building blocks, all these different opportunities to get through that design, build, test, learn cycle very quickly. And then they put them together in different ways until they came up with the final product, which is shown on the right. So moving from that, so we've got a prototype. How are we going to implement this? How are we going to get this up to scale? All through the technology development, we obviously have to be thinking about scalability. So we need to be able to scale up, we need to be able to scale down so that we can know that we're moving forward. And again, standardization comes into play, because it's once we get into implementation, once we want to put this into manufacturing, I need to do this hundreds of times. So it's not what can I do quickly, it's what can I do over and over and over again in a repeatable, high quality manner. So we shift our focus a little bit, but we still need some of the same principles in order to get to reliable product. And then in order to achieve value, I want to think about two things. One, achieving the broadest impact. So we think about the technology platform. It's really fun. We're engineers. We like to do that. But we really need to think about not just the technology and the equipment. We need to think about how does it fit into the manufacturing operation? How do we need to change the supply chain? What can we do from an environmental sustainability point of view in order to really get the most value out of these developments that we're driving? And as Mike mentioned, we need to manage risk in the space. So as you bring in any new technology, you bring in risk. In our business, there's always risk anyways. So managing that risk, not trying to eliminate it, not trying to pretend it's not there, but thinking about it and planning for it and addressing it is what we have to do. And that's things like supply chain dual sourcing, as Mike was mentioning. Knowledge management is actually, to me, a risk management technique. So knowing and building your knowledge and understanding how to access your knowledge, training your people, being able to connect your next development to your prior experience. Those are really important deliverables from knowledge management. So I would put that in that strong driver for knowledge management. And an example of this is how are you going to implement? And interestingly, of course, this is what Mike mentioned, scale out versus scale up. If I go all the way back to the learn from biology principles, if you look at how our cells work and how organisms are built, it's all a scale out technology. So we do not scale up one cell and make it bigger and bigger and bigger and bigger. We make more and more and more cells. And we do that even when we scale up into larger and larger bioreactors. And there's a cross point where the scaling out becomes so complex for us to manage that we do want to scale up. And so you can see that even though we're scaling out, Mike's now talking about scaling out at 2,000 to 4,000 liters. When we started in this single-use system conversation, we were scaling out at 500 to 1,000 liters. So we're scaling up and scaling out all the time, thinking about where that risk lives and what's going to give us the best productivity and the most reliability for our product. And you can see this came out of a very recent paper from thinking about tissue manufacturing. And you can see that, we're going from 2D to 3D. And as we go into 3D systems, which still require adherent systems, so you can see they either grow them as aggregates or as on microcarriers, we're starting to get into technology again. So their scale up technology in the advanced medicine of tissue regeneration looks a lot like our lab scale technology. And so how can we inform each other and move forward? And that brings me back to talking about single-use systems and advanced manufacturing and that cycle that we continually drive our abilities in biomanufacturing by advancing the technologies across the single-use space. So this level of information looks pretty familiar. So what do we need for advanced biomanufacturing? What do we need for gene therapies, for cell therapies, for tissue regeneration? We need all the same things we needed all along. We need a return on investment. We want to have the lowest possible capital investment to minimize risk, and we want to start it up as quickly as possible to get a value and revenue stream coming out of that investment. We need flexibility. We need to be able to convert processes from one to the other. Really important in some of these spaces where we're still learning about the product itself, that we're able to update and rapidly convert the process, which is, I would say, something that's a little different from our large scale operations with products we've been making for decades. And then we need to be able to expand rapidly. The amount of manufacturing that's needed for COVID, for example, if we start talking about mRNA as a new advanced biomanufacturing, where are we going to get that technology? If we talk about tissue regeneration, if you want to talk about COVID treatments, there's some really amazing work that there are some products that generate stem cells or mesenchymal cells that will treat the lung damage that's caused. And so we want to be able to expand these things really quickly. Operational efficiency really drives us to a smaller lot size with really fast turnarounds. But that, again, starts to, how does that trade off with this idea of expanding capacity? So we always are making trade-offs between these groups. And of course, we never trade off on safety and quality. And one of the things that single use really gives us in that space is totally eliminating the concept of cross-contamination, because we throw it away when we're done. So I would say that this single-use systems allows us to move forward. And as long as we continue to innovate in single-use systems, we will continue to enable more biomanufacturing, which will continue to pull from the single-use ecosystem more and more advances. So as we go through this cycle of innovation, it's really about how do we use single-use systems to move us forward from a biomanufacturing environment, and then how do we use biomanufacturing to push more development in single-use systems. And with that, I want to stop and I'll give you a couple of quotes that I like about the future. So if you don't push the boundaries, you'll never know where they are. And in the end, it's important to remember we cannot become what we need to be by remaining where we are. So I think it's really important. And the reason I chose these quotes is I really want to challenge the vendors, manufacturers, us in academia to continue to push development to make sure that we get the most robust single-use systems that allow us to keep moving medicine forward, because I think that's how all of us got started in this business and something we all want to contribute to. And with that, I'll wrap up and take any questions. And John, do you want me to stop sharing?
Thanks, Sue. So if anyone has questions, please put them in the Q&A or in the chat so that we can give opportunity to Sue to answer the questions that you may have. So I see one question. Do you guys at KGI provide training on single-use systems for your students?
We do. So our students, we have thanks to our partner Sartorius, we have a 50-liter subsystems that we use. We use single-use bioreactors at the 2 and 3 liter scale. We teach them our downstream operations, our bioseparations technologies right now are mostly the standard traditional systems, but we teach and use instruction in those spaces to talk about single use and introduce them to the concepts. They also do an internship in industry, which more and more they come back with having had hands-on experience. Our pilot scale in both the microbial and the mammalians include a lot of opportunity to access and use single use.
Great, thanks for that. What is considered the smallest volume when applying miniaturization with scope of upscalability?
So I think that is changing, and I think it's really, there's some really exciting things that are happening. I am a real big fan of a company called Berkeley Lights that moves things one cell at a time. And I think that if we get to the point of understanding things one cell at a time, we can bring them back together and use that information to properly manage the environment. When we do larger scale manufacturing, we put all these cells together and then we try to control that massive set of metabolic pathways with pH and temperature. And those are really our only dissolved oxygen. So we have a few very broad control parameters where we're trying to drive what's happening inside a cell. So it's really important, I think, as we get into these, that we can use these technologies that get us almost to the cellular level. And we have to learn how to take what we learn at that level and bring it forward. In terms of bioreactor scale, there's the Ambers at 15 and 250, and there's lots of multi-well plate-based systems as well. And I think it's a matter of learning your scaling parameters. It's a matter of learning how to scale down and then how to scale up in order to get effective data.
Thank you. So here's another question. What do you see as some of the current boundaries for single-use systems that we should be trying to push through?
So I think that the standardization is huge. We can't connect these things to each other sometimes, and I think that's really hard. And I think that being able to solve that problem is something I know the industry and the vendors are already working on, but I think it's really important that we're able to take the best technology and connect it to the next best technology. And so that level of understanding. Also, I think that the boundaries on reliability of our supply chains. Mike mentioned something that's happened in your supply chain in the last 6 months. I certainly don't know what that is, but I have experienced several of those in my time at Merck. And I think that ability to depend on suppliers means suppliers, it's a boundary that needs to shift into the supplier space. So suppliers need to start thinking about how do we make sure that this multitude of partners that we're supporting are getting the reliability of their supply chain. So that's one of the boundaries. Another is obviously continuous manufacturing. And I think as we look at technology development, how do we get more and more continuous in our production systems because of the efficiency that drives. And the efficiency really comes from the clock. So when we're running a batch operation that takes 20 hours to set up and 4 hours to run, we've obviously lost a lot of our time. And so getting to a continuous space and driving down the scale makes it easier for us to build these small modular standardized systems that then we can move maybe and distribute around globally and give a better global access to our products. I think those are some of my thoughts on the boundaries. I'm sure there's more.
Yeah, I think we'll continue to learn and understand more as we continue to push those boundaries, right, Sue?
That's right. That's right. And I think that drives the opportunity even further. So it allows us to push. And I think all of us are in this for the medical treatments, and it allows us to push forward and give more opportunities to our partners in the medical fields.
Sure. So as another question, do you foresee that there's potential for using 3D printing to create bioprocess equipment on demand?
I do. I think that we're still a little slow with 3D printers, but I think we're getting there. We're using 3D printing a lot for building scaffolds for things like stem cells and tissue regeneration technologies. So there's a lot of work going on there. We're actually have a research project going on at KGI looking at 3D printing of chromatography resins so that you could change the resin not on a macro level, but on a micro level, and control the exact dimensions as well as the configuration into things that might not be able to be achieved naturally. And I think there's opportunities around the board. One of the things that I always think is amazing potential is, you know, kind of using 3D printing as a blow, fill, seal type technology, where we print our containers as we go on the drug product space. But right now, I think the boundary there is the speed of those printers and the cost of the material of those printers. And I think that that's obviously advancing as well. And hopefully, if we can converge our ideas for how to use those technologies with their speed and their progress of developing that technology, I do think it's something that should be brought into the research labs. And once it's there, scientists get engaged and start thinking about how could I use this technology.
Wow, what a novel idea that you just gave us on make the container as you're filling it. That's true innovation there. I'm looking forward to seeing what that can look like. Yeah, we have to continue to think along those lines. That's not something I'd ever even thought would be something that we could do, but you're right, pushing the boundaries. Sure, why can't we do it?
And we do it with blow, fill, seal. So it's a matter of if we want rigid containers and we have different material needs than blow, fill, seal can do for us. How can we use this technology?
Nice. Do you consider large pharma as innovative or conservative? What would be the best approach to get more innovative?
I feel like in the last 5 years or so, there's been a shift and a lot more innovation coming out of large pharma, whereas it used to be wait for somebody else and then go buy that technology. And new technology, bringing in new technology, I've seen a bigger and bigger investment from large pharma. I think large pharma still has a huge legacy. There's a huge investment in legacy technologies that has to be continued, maintained, and how do we advance inside those legacy technologies? So if you already have a 20,000 liter stainless steel operation, how do you make it work the best it can work is really an other side of advancing. So I do think that there's room even in those spaces for advancing technologies without having to throw out and start over again. But of course, single use, and the reason we're here is it pushes us along those boundaries because it is so flexible and it is so much more amenable to being advanced. I think that that's really allowed the large pharmas to also start to push forward and take advances. The research operations are using single use, so they're because it's easier and more efficient for them, and they're able to hand that off to the development and then the manufacturing organizations. And I think that that's coming through and starting to really impact the manufacturing technology development at large pharma, as well as small pharma does it out of necessity. You know, startup companies, they start with these technologies as their basic platforms out of necessity. And large pharma is really starting to have that opportunity because they have both a foot in both camps, sort of. They can work in both technology platforms.
Nice. Thank you for that viewpoint. Another question. How are students of today different than when you were a student?
So well, it's a long time ago for me to remember when I was a student. I think that they're obviously a lot more technically savvy. The switch to virtual has been hard, but not nearly. Obviously, we couldn't have done it when we were in that space. I also think there's and this is probably a little bit driven by my role here at KGI, but I see a really strong focus on entrepreneurship and on thinking about how the business integrates with the technology. So our engineering programs are focused on, and we work in partnership with industry. I would say that when I was in academia, we were focused on what happened in the book and a little bit of what happened in the lab that was set up for us. And our students are so active and focused towards getting a research project and lab experience and coming to us with ideas about what we could do as a new project and bringing connections to us from industry, from their summer internships, or from, you know, even as undergrads, they come in with these industry connections and bring research to us. So I think there's a lot more engagement in that space.
Nice. Thank you. I would like to see if Mike has any questions for Sue. In your role in Merck with the MSDC, how do you see us, perhaps Merck, collaborating with Sue and KGI?
Sue, again, great talk. Really enjoyed it and really enjoyed the insights that you brought, different perspective that you brought. One of the things I'd like you to speculate on, if you would, is as we think about intensive use of single use, what does that do to the basics of facility design, both the process facility and the supporting quality laboratory facility?
So I think, so from the process facility first, there's a shift from needing large spaces for equipment to needing large spaces for storage of equipment. So there's it's a real thing to think about. Waste flow becomes a much bigger piece of the conversation because it's not happening in pipes. So I think it's, there's a definite, the facility size inside the classified space certainly can go down. And we have, we've developed technology around closed systems in all kinds of platforms, but allowing us to relax some of that classified space. But we still need big spaces for big tanks that are full of big piping systems. So we can get to smaller spaces from a process point of view, but we need bigger support spaces. And I think that's one thing that's often underestimated in designing a new single-use facility, is how big the warehouse needs to be, and how big the corridors need to be, and where you are going to have your hold for single-use buffers in single-use mixers, and they need to be able to be close to your system. So that's the impacts, I think, on the facility in the process space. On the quality space, there's, I think, an incumbent on the manufacturers to take a deeper understanding of the single-use equipment and treat them as supplies, and they need some level of incoming testing that's more than a C of A, that's somehow an understanding of, is this actually going to work? Is there a potential risk here that I'm not? And how deep you go on that, obviously you don't want to go so far that you're reinventing the entire quality system that exists at your manufacturer, because you trust your manufacturer of your systems, but you need to have some visibility as well, and some ownership of that in the quality labs. I could see also, though, more and more switching to how do we bring more single use into the quality labs, and how do we move from do we need to switch to single-use systems for prepping materials, for doing analytical techniques? These systems are developed at that scale to start. So bringing them into the quality lab would bring advantages to the quality environment as well.
Thank you, Sue. That was great. I just have one more question. In process development, oftentimes we think about, here are the unit operations I need. Here's what was traditionally done. Does it have a single-use version of doing this unit operation? And it strikes me that perhaps that's the wrong way to think. That maybe we ask instead, what types of unit operations are now possible with single use that were not possible before when dealing with stainless steel and glass? Would you comment on that?
Yeah, I think that's a really good observation. And I do think, if you think about where we came from, just the fact that you use the word unit operations means we came from, the 1940s and 1950s, right? Because this is the terminology of chemical engineering. And I think that the opportunity to go back, and that's why I like this framework that I found, to think about learning from biology. Go back and think about we're doing biology in most of these systems that we're talking about today. So thinking about, how does a cell solve this problem? How does an organism solve this problem? And then carrying that forward could potentially give us some new insight on some new ideas, and might give us advances and get us out of the space of, how does this look like a mixing tank? And how do I take this back to a Reynolds number, right? Because
right.
that's where we come from. And so I agree. I think that there's opportunity out there to be found, and it comes from going behind all of it, and maybe thinking about what's happening in that biology space. What are we trying to do? Is there a way to maybe get a cell to send us more pure material? I mean, it's something we think about, but from a cell line development point of view, we are spending more time thinking about the how to make more product, and can we think more about how to make it less other stuff? So we think about building in secretory pathways, but are there other things we could do? So yeah, I think there's room all the way back, and then that might inform us going forward.
Okay, thanks, Sue. Really appreciate it. Thanks.
Well, thanks for having me. It's really been great to catch up and to talk to you.
Likewise.
Thanks for those questions, Mike, and thanks so much, Sue. And thanks, Sue, for, let me share my video, my camera. Thank you for giving us things to look forward to, and new ways of thinking, of pushing the boundaries, and just a new way of thinking. As you pointed out to Mike, yeah, in the past, as chemical engineers, we've thought about unit operations, but you've given us a whole new way of thinking about looking at the cell level, what is it that the cells are doing. And thanks so much for your insight into 3D printing and perhaps where we can go with things like this. You even made a connection to the new Merck strategy of Expedition 2025 and how we need to take innovation and push the boundaries and look for new and unique ways of doing things within our industry. I liked what you were talking about with the 3 pillars of medicine and the design, build, test, learn philosophy. So thanks so much, Sue, for giving us things to think about and giving us an introduction to KGI. I look forward to seeing how we can collaborate going forward.
Great, and thank you so much. And I agree, I would love to find ways to collaborate more and continue to push out of unit operations into operations as we leverage technology and really as we look for what do we need and what do we have and filling that cycle around to make sure that we're moving things forward. I do want to say, you know, the sun came up while I was talking, so it's a great opportunity. And I did not go back and show you the final map of the where everybody is. So if you'd like,
I can please share.
I can do that and see if we. So I need to go back and share my screen so here we go. This is where you guys are today. Somebody in the Midwest, welcome. Lots of you on the East Coast, somebody down in Florida, people over in Europe. So there's where everybody is today.
Nice. Thank
you. Thanks again for allowing me to share this with you today and being part of the conference.
Thanks. So I also want to thank Mike. I thought this presented a very unique unique opportunity for Mike and Sue to come together and be able to share. Mike being able to introduce his longtime colleague of Sue and being able to collaborate. I think this is a great reunion and being able to bring other folks in Merck to have the same understanding and be able to learn from both Mike and Sue. Thank you so much for helping us kick off this year's event.